Hello everyone! Welcome to another episode of the Amino Bros channel. In this video, we will discuss how organisms constantly interact with their environment, responding to various critical stimuli in survival and adaptation. Stimuli are changes that can occur from our external or internal environment, such as light and heat, that causes reactions and responses that are meant to maintain homeostasis and adjust to changes. This reactivity is called irritability in animals, and it manifests itself in various behavioral reactions, such as taxis, which are movements toward or away from a stimulus. As simple invertebrates, earthworms present as an ideal model for studying these basic biological reactions. Their responses to stimuli can be analyzed to gain insights into the mechanisms underlying animal behavior and sensory perception. The learning objectives for this experiment are: first, to investigate the responses of earthworms to various stimuli, such as contact, light, heat, moisture, and chemicals. Second, observe how each body region displays varying sensitivities and analyze the direction of movement in response to stimuli so that people will gain an understanding of basic sensory responses in organisms. Lastly, deepen comprehension of environmental interaction and behavioral adaptation. The materials we use for this experiment are 2 dissecting pans, 1 damp sheet or sheets of tissue, 5 earthworms, dissecting needle, card with a 2 mm hole in the center, flashlight, xylol, alcohol lamp, and a lighter.
And now, let's talk about the procedures in doing the experiment, which will comprise the 5 taxis that we will try to emulate onto the earthworm. So, for the first procedure of the taxi is the contact. With the point of a dissecting needle, lightly touch the different body regions of the earthworm. And so, after 5 minutes, apply the repeated stimulation to the middle portion only. Next, for the next procedure is the light taxi. Using a card with a small 2 mm hole in the center and a flashlight, allow a spot of light to shine on the different regions of the earthworm, particularly the anterior, middle, and posterior. This we must observe if there will be any stimulation that happens by the earthworm or any reaction that might occur. Next is the heat. With a hand lens, or specifically the one we were given is an alcohol lamp, we will use a metal rod that we will heat with the alcohol lamp and slightly put the metal rod close to the earthworm so that we may simulate a big source of heat on the different parts of the body with it not touching the earthworm. Next is the procedure for moisture. Line a clean and dry dissecting pan with tissue paper. Dampen 1/2 of the tissue with water and leave the other part half dry. Place the worms at random over the entire surface, and after an hour or more, observe the location of the worms. Next is the chemical portion of the procedure. Moisten the tip of a glass rod with xylol and bring it close to the different body regions of the earthworm. Make sure the glass rod does not touch the worm, as it may mess with the potential reaction that should accurately occur on the earthworm.
Now here are the results of the experiment. The experiment utilized 5 different stimuli, namely touch, light, heat, moisture, and chemical. Let's start with touch. For its anterior part, the earthworm responded vigorously as the dissecting needle touched its anterior end. Next, for the middle part, the earthworm responded by wriggling, but with the least amount of movement compared to the other 2 parts. And lastly, the posterior end. The earthworm responded away, but with less rigorous movement compared to the anterior end. The next stimulus is light. For the anterior part, the earthworm moved away from the light more strongly compared to the other two parts. Next, for the middle part, the earthworm moved away, but with less intensity compared to the anterior part. And lastly, the posterior end, no movement or reaction or response was observed from the earthworm. The next stimulus is heat. For the anterior end, the earthworm moved away very quickly from the heat source, which is the hot rod or hot metal. For the middle part, the earthworm showed slight movement away from the heat source, and similar to the middle part, the posterior end also showed slight movement away from the heat source, but this movement is very minimal when compared to the anterior end. Next, the fourth stimulus, which is moisture. Comparing the two sides of the pan, which is the dry and the moist side, it was observed that almost all earthworms migrated towards the moist area. Only one earthworm remained in the dry area of the pan. And lastly, stimulus number 5, chemical. Xylol was utilized in this experiment. As the xylol was brought near the anterior, middle, and posterior ends of the earthworm, the following are observed. For the anterior end, as the xylol was brought near the anterior part of the earthworm moved away very rapidly away from the xylol. For the middle and the posterior ends, no response was gathered or observed. In relation to the results of the experiment, one question arises from it. Why did the earthworms respond that way in the presence of the given set of stimuli? This is where taxis and the earthworm's anatomy come to play. Taxis, which is by the way not the plural form of taxi, is an animal's directed movement towards, which is positive taxis, or away, also known as negative taxis, from a certain stimulus. In the experiment, 5 different stimuli were introduced to our earthworm, namely touch, light, heat, moisture, and chemical. This means that 5 different types of taxis were involved and observed from our earthworm sample. Let's start with the first stimulus, which is touch. In the experiment, a negative response was observed from the earthworm as the dissecting needle touched the anterior, middle, and posterior parts of the worm. But why did it give that response? Earthworms have epidermal receptors in their skin that helps them detect the presence of touch, temperature, and some chemicals. These epidermal receptors are located and distributed within the epidermis of the earthworm, showing a slightly elevated cuticle that covers groups of receptor cells. These receptor cells have hair-like processes in their outer ends, while the inner ends are connected to the nerve fibers. That is why when the receptor cells are stimulated, the earthworm's movement away from the touch was observed as its response to the stimulus. The second stimulus is light. Unlike the epidermal receptors, photoreceptors in earthworms decrease as you move towards the posterior end of the organism. These photoreceptors are more evident in the prostomium and peristomium areas of the earthworm, which are found at the anterior part of the organism. That is why a reaction to light, specifically a negative response, was only observed in these areas during the experiment. Additionally, the presence of light often indicates the presence of heat, for example, sunlight. Hot environments are not suitable for earthworms because they might die because of it. Earthworms breathe through their skin. During the process of respiration, earthworms release internal fluids, allowing them to trap oxygen and use them for breathing. Too much heat will cause these fluids in their skin to dry out and eventually lead to their death. That is why during the experiment, when the hot rod was brought near the worms, a negative response was observed from them. Moreover, this is the reason why in the fourth stimulus, which is moisture, the earthworms migrated towards the moist and damp area of the pan. Having a moist environment allows them to survive, thus resulting to the response or reaction that they showed during the experiment. And lastly, number 5, chemical stimulus. Xylol was utilized in this experiment and the earthworms showed a negative response in the presence of said stimulus. Earthworms have buccal receptors found in the epithelium of their buccal cavity. These buccal receptors are responsible for detecting olfactory, which relates to smell, and gustatory, which relates to taste, functions or processes. Given that xylol is a toxic, irritating, and foul-smelling chemical, close contact of the earthworm led to the worm's chemoreceptors to reject the chemical, resulting in the negative response observed in the experiment.